We'll now begin the section on protein processing. After proteins are translated, they're often modified in a variety of different ways. And the reason that they're modified is fourfold. One is that modification of the proteins can increase their stability or decrease the degradation of the proteins. Or in certain cases, modification of the proteins decreases the stability and allows them to be turned over at a higher rate. Another function of modification is to target the proteins to particular locations, either within the cell or outside the cell. The third is modulation of activity. Processing proteins in different ways can change or regulate their activity. The fourth reason for modification is to increase or decrease the solubility of proteins. Some examples of protein modifications are shown below. These include things like phosphorylation, glycosylation, hydroxylation, peptide cleavage, protein cross-linking, loss of the N-terminal methionine on a protein, acylation, which is the addition of fatty acid groups, addition of prosthetic groups, loss of the signal sequence for membrane proteins or for secreted proteins, or ubiquitination, which regulates the stability or leads to the degradation of proteins. We'll talk about a few of these in more detail. The first of these is phosphorylation. Phosphorylation is the process by which phosphate groups are added to proteins. And there are two enzymes which are responsible for this. The enzymes which phosphorylate proteins, that is, add phosphates to proteins, are called kinases. Those that remove phosphate groups from proteins are called phosphatases, which cause dephosphorylation. Now, phosphorylation, or addition of a phosphate to a protein, can change the conformation and or the activity of these proteins, either leading to increased or decreased activity. Phosphorylation, then, is a major mechanism of signal transduction in which we can regulate the function of proteins. And as we go through biochemistry, you'll see lots of examples of how biochemical processes are regulated by phosphorylation. Glycosylation is the process by which carbohydrates are added to proteins. Now, glycosylation occurs in the endoplasmic reticulum or in the Golgi apparatus, and particularly occurs to proteins which are being secreted from the cell or targeted to particular intracellular compartments. The function of glycosylation is threefold. First, glycosylation may increase the solubility of proteins. This is because the carbohydrate or sugar groups which are added to the proteins are highly hydrophilic, and this allows the proteins to dissolve more easily in water. Glycosylation may also protect proteins from proteolytic degradation, extending the half-life of these proteins and causing them to be more stable. Glycosylation is also important in the proper targeting of proteins to the correct functional locations, either to intracellular organelles or to be secreted from the cell. Glycosylated proteins are classified according to the relative proportions of protein and carbohydrate in the protein. Glycoproteins are made mostly of protein, with a few added carbohydrates. Proteoglycans, on the other hand, are primarily carbohydrates that have been added to a protein backbone, is illustrated below. Glycosylation is one of the major mechanisms by which the cell designates the eventual location or destination of a protein. And this is the mechanism of protein trafficking, or moving proteins from one location to another in the cell. An example of this is lysosomal trafficking. Lysosomes are membrane-bound organelles that contain hydrolytic enzymes responsible for degrading proteins and lipids and carbohydrates and other things in the cell. These enzymes are regulated by pH. That is, they're only active in the low pH of the lysozyme, and this protects the cell by only activating those enzymes in a specified location. If they were activated in other locations in the cell, they would degrade proteins and lipids and carbohydrates in places that they're not supposed to, and that would cause the cell to die. So how do we ensure that these proteins make their way to the lysosome and not some other location? Well, the way that's done is that after translation, these proteins are glycosylated with a sugar that we call mannose-6-phosphate. This occurs in the endoplasmic reticulum of the cell. There's a receptor in the Golgi apparatus which recognizes and binds the mannose-6-phosphate groups and causes these proteins then to be sequestered in transport vesicles which carry them to the lysosomes. Once in the lysosomes, then these proteins are activated by the low pH environment there, and they can start functioning, that is, in degrading lysosomal contents. This is schematically illustrated in this picture. You can see a lysosomal hydrolase which has been translated in the endoplasmic reticulum. You can see as it moves to the Golgi apparatus that a mannose-6-phosphate group is added to it. In the Golgi apparatus, then there's a mannose-6-phosphate receptor which recognizes that mannose-6-phosphate group, causes that protein to be sorted into a vesicle, which then carries that to the lysosome. In the lysosome, then, the protein dissociates from the mannose-6-phosphate receptor, and it begins to function in the lysosome. The mannose-6-phosphate receptor is recycled in a vesicle, then back to the Golgi apparatus, where it can pick up additional enzymes and carry them to the lysosome, and that cycle is repeated over and over again. Now, this mechanism can go wrong and lead to a particular disease, which is described in the following clinical vignette. A 6-month-old infant presents to his pediatrician for failure to thrive. When you do a clinical history, you see that he is not reaching his developmental milestones. He's very small for his age, and his mother reports that he's had very frequent upper respiratory infections. Here's a picture of what he might look like. When you do an initial physical examination, you see that he's very abnormal. He has very coarse facial features, including a high, narrow forehead, flattened nasal bridge, and a long philtrum. You also notice that he has macroglossia, or a large tongue, and he has hyperplasia of his gums, what we refer to as gingival hyperplasia. His hips are loose, they're easily dislocated, but the other joints show restricted movement, don't move as well. When you do a neurologic examination, you notice that his muscles are markedly hypotonic. Laboratory testing detects deficiencies in several lysosomal enzymes, including hexosaminidase, iduronate sulfatase, and aryl sulfatase A, when you culture fibroblasts from this patient's skin. What is the disease that this patient has? Well, this patient has a defect in lysosomal trafficking, which we call eye-cell disease. Eye-cell disease was described in 1967 in a patient that had signs and symptoms similar to Hurler syndrome, but with an earlier onset. Biochemical studies of cultured fibroblasts from these patients showed that the cells were deficient in a number of lysosomal hydrolases. Instead, the enzyme activities were detected in the culture medium instead of in the cells where they belonged. So if you look at that information, what do you predict that the biochemical defect will be in these patients? Well, eye-cell disease is caused by deficient targeting of lysosomal hydrolases to the lysosome. These hydrolases are synthesized normally, and the mannose-6-phosphate receptor is present. However, there's a deficiency in a particular enzyme, which I've listed there, a phosphotransferase, such that the mannose-6-phosphate group is not added to the lysosomal proteins. The absence of this mannose-6-phosphate means that the mannose-6-phosphate receptor does not recognize those proteins as lysosome-bound. So what happens then is these lysosomes get secreted rather than being transferred to the lysosome. Now, that means that the lysosome has no hydrolases, and the absence of these lysosomal hydrolases causes the accumulation of various materials in the lysosomes, leading to cell death, especially in the nervous system and in the skeletal system. The lysosomes that have accumulated material in them appear as inclusions in the cells, and so this disease acquired the name inclusion cell, or eye-cell disease. Here's a schematic of what happens in this disease. You can see that the lysosomal hydrolases don't have a mannose-6-phosphate group, and so they're not recognized by the mannose-6-phosphate receptor. So instead of being sorted into vesicles which will carry them to the lysosome, they're sorted into vesicles which fuse with the plasma membrane and cause these hydrolases to be secreted into the extracellular fluid. As a consequence of that, material accumulates in the lysosomes and forms inclusions in the cells. The signs of eye-cell disease include facial and skeletal abnormalities, characteristic coarse facies, as you saw in that picture, gingival hyperplasia, and mental and developmental retardation. Children who have this disease are often afflicted with frequent upper respiratory infections, and the course of the disease is characterized by progressive psychomotor deterioration, leading to death in early childhood. There is no known treatment for this disease. Let's discuss now proteoglycans. As I talked about before, proteoglycans are molecules which consist of a protein backbone onto which numerous long chains of carbohydrates are added. Now, these proteoglycans are especially important in the function of the extracellular milieu. The proteoglycans make up the amorphous ground substance component of the extracellular matrix, and they have important functions in particular tissues. For example, in cartilage, where they serve as a cushioning material to lessen compressive forces on the joints. Now you can imagine how that would function. Because these have so many carbohydrates on them, they're very hydrophilic. They attract and trap water, causing them to have a cushioning type consistency, and that functions very well when you need cushioning effect, for example, in your joints. So what's the difference between a proteoglycan and a glycoprotein? And so here's a picture of what these might look like. You see the hyaluronic acid as a long molecule to which there are proteins attached. These proteins attach the proteoglycans to this hyaluronic acid molecule. And you can see that this creates a lot of surface area with a lot of carbohydrate that attracts a lot of water. And that's what provides the function of these molecules in the extracellular matrix. Now, some of the common proteoglycans are names that you probably recognize. These are things like hyaluronic acid, chondroitin or chondroitin sulfate, dermatan sulfate, heparan sulfate, heparin, and keratan sulfate. And these have functions in the extracellular matrix of different tissues in the body. Now, these molecules are synthesized by taking individual sugars and adding them to growing chains by UDP-bound intermediates. The degradation of these is done by specific enzymes which remove each of these sugars sequentially. Some of these enzymes have names which correspond to the particular proteoglycans which they degrade. For example, iduronate sulfatase or iduronidase, heparan sulfatase or glucuronidase. Now, these degradation enzymes can also be defective in certain inherited disorders, an example of which is shown in the following clinical vignette. An 18-month-old boy is brought to a clinic by his mother. She's concerned because his abdomen has become increasingly distended over the past 6 months. He doesn't seem to be growing normally. He was average height at his 9-month checkup, but after that his growth has progressively slowed. When you measure him, you notice that he really is growth retarded. Physical exam reveals coarse facial features, clouding of his corneas, and hepatosplenomegaly, which accounts for the abdominal distension described. A liver biopsy is performed and shows distended hepatocytes with cytoplasmic clearing. When you stain these with a PAS stain, you notice that these are full of PAS-positive material. Here's an example of the corneal clouding which you might see in this disease. So what does this patient have? This patient has a disease called Hurler syndrome, which is also called mucopolysaccharidosis type 1. Hurler syndrome is an autosomal recessive disease that is in the general category of mucopolysaccharidoses, or MPSs. The MPS syndromes result from deficiencies in these enzymes that are responsible for degrading proteoglycans. Some of these enzymes we described before, and they're associated with specific syndromes. For example, alpha-L iduronidase is associated with Hurler syndrome, iduronate sulfatase is associated with Hunter syndrome, and beta-glucuronidase is associated with Sly syndrome. The pathologic features of this disorder all result from the accumulation of undigested proteoglycan material in lysosomes. And so this falls in the general category then of lysosomal storage diseases. And like most of the lysosomal storage diseases, the accumulation of this material in lysosomes leads to progressive cell death. The specific clinical features of Hurler syndrome include growth retardation, hepatosplenomegaly, coarse facial features, and corneal clouding, as we described before. The organ damage in these particular diseases is most pronounced in the liver and spleen, as it was in this patient, in the bones, and in the brain, which causes the neurologic findings that we saw in this patient. In addition, you may develop subendothelial deposits in blood vessels, and this leads to cardiovascular complications, can lead to clogging of the coronary arteries, leading to very early heart difficulties. And death usually occurs by the ages of 6 to 10, and usually as a consequence of these cardiovascular complications. Now, mucopolysaccharidosis can be treated successfully by bone marrow transplant. That is because the transplanted macrophages that come from a donor actually produce the enzymes which are necessary to degrade proteoglycans, and so you don't get the accumulation that you see in a patient with the disease. Hunter syndrome is very similar to Hurler syndrome, but it's much less severe. It exhibits X-linked recessive inheritance, and you can remember this by a little mnemonic. Remember that Hunters shoot for the X, and that'll remind you that Hunter syndrome is an X-linked recessive disorder. SLY syndrome shares many of the clinical features of Hurler syndrome, but including its autosomal recessive inheritance, but they also may have inherited skeletal deformities which are characteristic of that disease. To diagnose these diseases, you have to do specific enzyme assays or genetic tests looking for the defect in these enzymes. This concludes the section on